In the last screencast, we talked about some basics of membrane transport. We talked about diffusion, concentration gradients, what concentration actually means, and a little bit about osmosis. Now we're going to talk about something a little bit more complex, which is osmosis in the cells. As you'll recall from class, osmosis is a type of diffusion, and it's just going to be the diffusion of water through a selectively permeable membrane, which we'll abbreviate as SPM. And in osmosis, we have the water going from an area of high concentration to an area of low concentration. We're focusing on a particular substance, which is water, and this is going to deal with living things, so we need to think about the plasma membrane as that selectively permeable thing. We're going to talk about three specific scenarios. First, we're going to look at osmosis in something called a hypotonic solution. Then we'll look at osmosis in a hypertonic solution. And finally, we will talk about osmosis in an isotonic solution. Oops, that's the wrong color. So let's get into this first scenario. So here's a beaker with some water in it. And we're going to say that this is distilled water, so it's 100% water, nothing else. And right now it may seem a little weird for me to say that there's 0% of anything else, but you'll understand why in just a second. So then we put a cell, drawn in pink there, into the distilled water, and cells aren't made up of 100% water. Let's say that this is 90% water and 10% everything else with all of those organelles. And remember that this pink circle, this lining around the cell, is going to be the selectively permeable membrane, the plasma or cell membrane. And that means that we're going to have movement in and out of the pores of the plasma membrane. So this is a hypotonic solution because the cell is hanging out in an environment that is hypotonic to itself. So that means that there's a lower concentration, okay, forget where it was, but it means that we have a lower concentration of solutes than itself, the cell. So 10% solute within the cell and a 0% concentration outside of it. So hypo means less than, and tonic is going to mean concentration. And remember that this is referring to what's outside of the cell, right? And we're always talking about the solution, not the cell. That would be more complicated, right? And the only thing that can move in this case is going to be water. We know it has the pores there. And the selectively permeable membrane won't allow for the movement of other solutes. So if no solutes can move, then water is going to be the only molecule that can move. And we still have a water concentration gradient. We have 100% water outside the cell, and then we have 90% water inside the cell. So there's a higher concentration outside than inside. So there's going to be a net movement of water into the cell. And I'm showing you that with these red arrows. Water moving into the cell, going from an area of higher concentration to lower concentration. And this is going to be like blowing up a balloon. The cell is going to expand in size. When a cell gets bigger due to water flowing in through osmosis, we say that the cell is going through something called cytolysis. Or we say that the cell has cytolyzed. And just like a balloon, if we blow it up too much, it can burst. And when a cell actually bursts, we call that lysing. Or we say that a cell lyses, or it's undergoing lysing. And that happens when there's too much osmotic pressure, too much pressure inside of that cell. And this is an important concept to think about when you get shots, for instance, like the flu shot. Because if the flu shot was in plain distilled water, like we have in this situation here, it would actually cause your blood cells, like we have shown here, to actually burst or to lyse, because they would expand a lot and cause them to burst open, and you would see a lot of bruising, which is not a good thing. So shots are actually in a saline solution, and that means that the shots are going to be balanced with the concentration of water that you have in your blood. So the second situation is a cell, which we're going to draw in pink here, in a hypertonic solution. So the cell is surrounded by this hypertonic solution, which means that it's going to have a higher concentration of solute outside than in the cell. So for this example, let's say that outside of the cell we have 80% water and 20% solute. I guess it could be sugar, it could be salt, whatever. And then inside the cell, we're going to make it have a higher water concentration, say that it has 90%, and let's say, obviously, then 10% of other solute. So the solute has a higher concentration than the cell. And the solute would want to pour into the cell, but it can't because of this selectively permeable membrane. Let's label that again, SPM. The only thing that can move is water through the pores, and water has a higher concentration in the cell than outside, so the water will then move from an area of high concentration to an area of low concentration. When you're going through examples like this on a quiz or test, I strongly suggest that you mark for yourself where is the area of high concentration and where is the area of low concentration. And I'll tell you if the membrane can allow certain things or not, but we know that this is the high concentration, this is the low concentration, so we then know which direction the water can flow if we're talking about osmosis. So when water is going to leave the cell, then it will shrink in size. The cell will sort of look like that. And the cell will shrink until the concentrations are the same inside and outside of the cell, where it's reached equilibrium. And when you have this issue happening and you have cells shrinking in size, we call this plasmolysis. Or the cell is going to, cell plasmolyzes, right? It won't implode, but it will get really small and it'll kill the cell if it doesn't get enough water. This is an example of, if we're thinking about real world, this is what's happening when a flower wilts. The cells are shrinking and overall the whole plant wilts as well. Let's move on to our third situation. We have that concentration outside of the cell, and we're going to have the exact same concentration, in the cell. This means that we're in an isotonic solution. The prefix iso means the same. So if I were to ask you, will we have plasmolysis or cytolysis here? You should be able to say, no, we're not going to have any net movement of water. So neither plasmolysis nor cytolysis will occur. So we've talked about hypotonic solutions where the concentration is going to be higher outside and lower inside, so we have the water flowing in. We have hypertonic solutions where we have a higher concentration inside the cell and lower out, so the water flows out. And then we have isotonic solutions where there's no movement because the concentration is the same inside the cell and in its surrounding solution. That's it.